In this video, we're going to talk about antibodies. So antibodies are produced by activated B cells during the humoral immunity response. And these activated B cells are actually, it's a specific type of activated B cell called the plasma cell. So these plasma cells secrete these antibodies, otherwise known as immunoglobulins, in response to coming into contact with an antigen. And so they bind to these pathogens or bind to these antigens once the antibodies are made, and once they bind to them, they basically flag those pathogens for destruction. So that will essentially tell the immune system this is something that's foreign and this is something that we need to destroy and remove from the body. So let's look at what these antibodies look like. So this is an antibody right here that is produced by those plasma B cells, and you'll notice there are two separate regions to the antibody. The lower region is called the constant region. So you can see it's colored in teal and purple here. And so this constant region is going to be the same for every antibody in that same class. So this is what determines what class the antibody is in. So whether it's like IgA or IgG or IgM, that's determined by the constant region down here at the bottom. Now at the top, you'll notice what's called a variable region. This is the region that differs for every single antibody that our cells produce, and the variable region is going to be determined by the antigen that it binds to. So you'll notice that there's a shape up here at the top, and that shape will correspond to the shape of the antigen that it will be able to bind to. So each variable region will look different for every single antibody that our plasma B cells make, because each antibody will need to be able to bind to different types of antigens, right? Whether it be an antigen from a Streptococcus bacteria or Staphylococcus bacteria, those may have different markers on them, different structures, and so those variable regions will need to be differently shaped in order to be able to bind to those antigens correctly. So we can take all these antibodies and group them into 5 major classes. We have the IgA, IgD, IgE, IgG, and IgM antibodies. So starting with the IgA, these are the antibodies that are found mostly in your bodily fluids. So you're going to see these a lot in mucous membranes. We see them in tears and saliva, things like that. These types of antibodies are also the ones that get passed from mother to offspring. So during pregnancy, the IgA antibodies can actually cross the placenta and pass from the mother to the fetus, and that will temporarily help protect the fetus from antigens that the mother potentially was exposed to during pregnancy. Once they are born, at least for a few months, these IgA antibodies can also be passed from mother to baby through breastfeeding. So you can pass these antibodies through breast milk as well. IgD antibodies, these are ones that are found on the surface of your B cells. So we said that B cells have membrane receptors on them that allow them to be able to bind to antigens and respond to those antigens by either producing producing antibodies or some other type of function. But in order for them to be able to respond to those antigens, they have to be able to bind to them first. And so those receptors on their cell surface are made of these IgD antibodies. That's what they are. That's what they look like. They're going to look like the IgD antibodies. IgE antibodies are typically ones found on the membrane surface of basophils and mast cells. Both of these cells are involved in producing histamines, and they're going to be highly involved in allergic responses and hypersensitivity responses to allergens and other types of antigens. This antibody IgE class is also going to be involved in helping us defend ourselves against parasitic infections as well. Now, the IgG class is going to be responsible for most of the adaptive responses or the adaptive branch of the immune system, which means it's going to make up most of the antibodies that are circulating through our body at any point in time. So about 80% of the free circulating antibodies that are found in our body that are there to bind to pathogens are IgG antibodies. Another type that's going to be kind of freely floating through the body or freely circulating are the IgM antibodies. These are a little bit larger and more complex looking, but they're going to serve the same purpose primarily as the IgG. They're going to help bind to antigens in the body and allow us to be able to respond to them using our adaptive responses. Now, IgM antibodies are also going to play a role in agglutination responses. So when we talked about how, when you're donating a blood sample to someone, if your blood types do not match, it would cause agglutination. That's due to those individuals' IgM antibodies potentially binding to antigens on the surface of the other person's red blood cells that they've received the blood donation from, and that's what causes that agglutination reaction, is those antibodies binding to those cells and causing them to clump up with one another. So IgM antibodies are going to play a role in agglutination. They're also going to help play a role in activating complement. We talked about those complement proteins in this chapter, which are used to insert themselves into the cell membrane of bacterial cells, and they can help essentially poke a hole in that bacterial cell to kill it. IgM antibodies play a big role in activating those complement proteins and showing them what antigens they need to actually bind to and insert themselves into the membrane of to help destroy. So overall, the human immune system is capable of making at least 10 billion antibodies, and sometimes even up to 1 trillion different types of antibodies, which is important because we need to have great diversity in these antibodies so that we can respond to the great diversity of antigens that we have around us at any point in time throughout the day.